Okay, so we talked about organic compounds in class the other day, and now we want to spend a little bit more time talking about the individual macromolecules. So today we're going to talk about carbohydrates. So carbohydrates name says something about its chemical makeup. Carbohydrate. So a carbon bonded to water. So the basic formula for carbohydrates is CnH2O to the N. So in multiples of. So this could be C6H12O6, or it could be C3H6O3. So that's what that refers to. So you can see it's made up of carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. So if you had to identify a chemical compound and all you saw was carbon, hydrogen, and oxygen, count up the ratio between your hydrogens to oxygen, and you should have a 2 to 1 ratio in order for that to be a carbohydrate. So in 9th grade, we talked about the functions, and we did a pretty good job with that, I think. So the functions of carbohydrates is simply to provide energy or to store energy. It's also used as a structural component inside of cells. So sugars can be found as monosaccharides, disaccharides, polysaccharides. So they might exist all by themselves, or they might exist bonded to other sugar molecules. They exist as chains, or they may exist as rings. So you'll see either type of structure as it relates to carbohydrates. Notice that we count the number of carbons here. So that's new to us. We didn't do that before. You want to start up here at the carbonyl group, and then you're going to count 1, 2, 3, 4, 5, 6, however many there are, straight down the chain. Over here, you kind of have to identify where was that carbonyl group, and then start counting from there, and then go around clockwise. So here you can see is where the original carbonyl group was, but for it to fold back upon itself and form a ring, it's going to break that double bond and form a single bond between this carbon atom and this oxygen atom. So over here, we start with this carbon, 1, 2, 3, 4, 5, 6. And so that's how we count them. I've also heard it explained as the carbon with the most mass kind of attached to it is going to be your number 1 carbon. So this carbon has 2 oxygen molecules, so that automatically has more mass than any of the other carbon atoms, and so that's going to be your number 1 carbon, 2, 3, 4, 5, and 6. Sometimes in organic chemistry we kind of simplify structures and we don't even put the carbon atoms in here. So we might just draw out this shape and we assume where 2 lines come together there is a carbon atom attached. So this is just a close-up of that numbering of carbon atoms. So again, you want to look for the carbonyl group in the stick figure here. The carbonyl group is at the top, so this would be your number 1 carbon, 2, 3, 4, 5, and 6. And we say the first prime, the second prime, the third prime. And that's going to kind of come into play when we start bonding molecules and we want to say where the bond occurred. So that's going to be useful. Also, when we talk about nucleic acids, we'll talk about the 3' carbon and the 5' carbon. So you really need to know what that number is referring to. Again, over here, you have to figure out where was that carbonyl group. So this carbon has 2 oxygens attached to it, which means this is where we originally had a double bond, and now the double bond has been broken in order for the carbon to the sugar to fold back upon itself. So this is the carbon with the most mass, so this is number 1, and we'll go clockwise, 2, 3, 4, 5, and 6. So saccharides can exist as monomers, or they can exist as polymers, so many units or a single unit. We would say saccharide if we're referring to any sort of sugar, okay? So monosaccharide, 1 sugar. Notice you're just going to have the basic CH2O to the N. A good example, and we'll use this a ton, is glucose. Glucose is a blood sugar, so you know the majority of our energy is coming straight from glucose. So we should be able to rattle off its formula, C6H12O6. We should recognize this hexagon as being glucose. That you want to get used to seeing and just knowing what it is. Sometimes you have two sugars bonded together, and there's many examples, but I'm just going to give you a very common example. So sucrose, which is table sugar, made from sugarcane, is a disaccharide. So you have a glucose molecule bonded to a fructose molecule. So fructose from fruit is also a monosaccharide. So the two of those together make table sugar. And then we can bond lots of glucoses together to make cellulose. Cellulose, you know, is in the cell walls of plants, so it serves as a structural component. And that is made by bonding many sugar molecules together. So some examples of monosaccharides are glucose, blood sugar, fructose, fruit sugar, and galactose from milk. Some other common examples that you should be familiar with from DNA and RNA are deoxyribose and ribose. Some characteristics of glucose is that they contain at least two hydroxyl groups, so you see more than two hydroxyl groups over here. They also have an aldehyde or a ketone. Do you remember the difference between an aldehyde or a ketone? So this one is on the end, so that's an aldehyde. Like A is the end of the alphabet. This one here is on the second carbon, so it's somewhere in between. So this is a ketone, and I think of K being in the middle of the alphabet somewhere, so ketones are in the middle of the molecule. So you'll notice that both glucose and fructose have the same chemical formula. If I counted up those atoms, I would have C6H12O6. So how are they different? How do they have different properties? They're isomers. Remember, isomers are molecules that have the same chemical makeup, they're just arranged differently. So you'll notice that the carbonyl group is on the first carbon versus here it's on the second carbon right there. Okay. Other characteristics of sugars are that they're sweet to taste. This probably satisfies our need for carbohydrates, right? Because we like sweet things, so that ensures that we're going to eat them. And we need them for structural components and also for immediate energy. Another characteristic of saccharides are that they dissolve readily in water. So would that make them polar or would that make them nonpolar? Probably polar. So water is going to attract pieces of them, right, and pull them apart, and so that allows them to dissolve. Disaccharides, as the name would imply, are two sugars. So here's a couple common examples of disaccharides. You have sucrose, lactose, and maltose. So sucrose we already talked about. Sucrose is glucose plus fructose. Lactose is also found in milk, glucose plus galactose. And then maltose found in malt, you know, like malted milk. I love shakes. So that's glucose plus glucose. All of these taste sweet and dissolve easily in water. So that goes to that hydroxyl property. Sucrose is the most plentiful sugar in nature. I don't know if you knew that, but it's an interesting tidbit. In humans, glucose circulates as blood sugar, but in other animals, it's a disaccharide circulates instead of the monosaccharide. Sometimes you hear oligosaccharide instead of polysaccharide. So this oligo prefix is referring to few. So they consider a few somewhere between 2 and 6. So between 2 and 6 sugars bonded together, monosaccharides, and you have an oligosaccharide. The most common oligosaccharide, the one we would be most familiar with, is the oligosaccharide that is found, kind of like an antenna here, in your cell membrane. So it serves in cell communication, cell-to-cell recognition. Okay, so dehydration synthesis we talked about the other day. It's also known as condensation. So recognize either term. Sometimes a question might throw in both words as an answer choice, in which case probably neither one of them are the correct answer because they are the same thing. But sometimes it might be the answer and you're looking for dehydration synthesis, but instead they give you condensation. So you want to recognize that that is the same word. So we talked about how we dehydrate a molecule by taking a hydroxyl off of one and a hydrogen off of the other. So those two will come together and form water, and then the two molecules that are left will form a glycosidic bond, and that glycosidic bond then holds your two monosaccharides together to make a disaccharide, or you might have a bunch of disaccharides bound together and you have a polysaccharide. So we're going to do an activity in class where we get to make some polysaccharides. The only point I'll make here is that each of these chemical reactions that happen in our bodies require enzymes. So anything that can affect an enzyme's function, like pH, temperature, or concentration, will affect the chemical reaction. So these dehydration synthesis, or if I had to break this in, it's a hydrolysis reaction, either way, I'm going to use an enzyme in order to make that happen. So for each of the polymers that we make, we're going to use a different term to refer to the bonds that are being made. So this that we formed here is called a glycosidic linkage. So in our sugar molecules, we'll use glycosidic linkages. And hopefully you recognize this glyco portion as looking similar to glucose. So that should help you remember that those are found in saccharides. Now there's two different types of glycosidic linkages, and we're not going to test you on this, but you have an alpha bond and you have a beta bond. So the alpha bond is a cis and the beta bond is a trans. And the difference is that, well, you can see the position of the bond between the first and the fourth carbon atom. Remember, we got rid of a hydroxyl group and a hydrogen atom. So they were taken from the same position. But over here, these are stereoisomers of each other, so they look alike, but maybe in a mirror image. So in this case, the hydroxyl group came from a different position. And so it creates a different bond. And this is going to have a higher boiling point, and it's going to give the molecule different structural properties. So in my class, we already had an introduction of condensation and hydrolysis. So just a reminder that breaking of those bonds is going to be a hydrolysis reaction. So in order to break this glycosidic linkage right here, we're going to have to pretty much shove a water molecule in there because we took one out in order to bond them. So hydrolysis, breaking with water, or water breaking, you'll take a water molecule and it splits into a hydroxyl and a hydrogen atom, and they join on the first and fourth carbon atoms, and then you've broke the molecule apart. So where does this happen in a cell? Just making some other connections. It happens in the lysosome. So lys, remember, means to break down. Some is body. So this is a breaking down body inside of the cell, and that's its job. Okay, so this is where the alpha and the beta glucose comes into play with polysaccharides. So that position of the bond is going to be really important. So notice these two alpha molecules are energy storers, starch and glycogen. We can break them down and release energy from them, right? So that's a pretty easy bond to break. Remember, I said the beta glucose has a higher boiling point, and so that means it's going to be harder to break bonds, right? So cellulose and chitin both are more difficult to break apart, so doesn't it make sense that we would use those for structures rather than energy, because we need to be able to break things apart to release energy. So again, making that connection between structure and function. Do you need to memorize which is alpha, which is beta? No. But is it good to recognize that the different shapes give it different properties? Yes. So you should be able to explain that. So I think you know starch is stored in plants. Glycogen is stored in liver and muscle cells of animals. So that's where we get our backup storage of energy. Cellulose, you know, is in the cell walls of plants as well as algae. You might have thought algae is a plant, but it's a protist. And then chitin is in the cell walls, if you will, of fungi, and it's in the exoskeleton of arthropods and some other animals like crustaceans. So here's the alpha and the beta glucose molecule that we're talking about. Notice this first position, the first carbon atom. The hydroxyl group is in a down position for the alpha, and it's in an up position for the beta. So if you were to bond two alpha molecules, notice the two hydroxyl groups that will be bonding are both in the down position. So that's a pretty easy bond to break. Whereas if you were bonding two betas between the first and the fourth, notice it's kind of an up and down bond. So that is a sturdier bond and not as easy to break. So continuing on with that beta glucose, we're going to look at cellulose. So here you see the beta glucose position, and and every other molecule there, right? So, or every other monomer that makes up the polysaccharide. So the kind of cool thing about that is it allows these molecules to bond with each other. And so it can be laid down into plates or layers. And so that's why it serves a structural purpose in both chitin and cellulose. So you can see how these can be attracted to each other and form bonds between the hydrogen and the oxygen molecules of two different strips of glucose. So that makes them pretty sturdy in the cell walls. Cellulose is the most abundant compound on Earth. Makes sense. It's in plants, and plants by number is the greatest amount in our food chain, right? Bacteria probably is a higher number overall, but they're pretty small. So herbivores, like cows, have a way of digesting cellulose, but we do not, because we can't fit this beta glycosidic linkage. We don't have any enzymes that fit that. And cows have these wonderful bacteria living in them. So bacteria is pretty awesome, that are able to digest that cellulose for the cow, so the cow can still get nutrition from the grass. So again, we break polymers with hydrolysis reactions and enzymes. So we're going to have to split this water molecule into a hydroxyl group that will bond on the 4th position, and a hydrogen atom that bonds here in the 1st position, according to this picture. So enzymes will fit. They fit the cis, for us, they fit the cis bond where they're both in the same position, but they don't fit the trans bond, which is when one is up and one is down. So we've looked at hydrolysis reactions before, so I think we understand how that works. So this is just a bigger picture of that bond. This is the alpha linkage. This is considered cis because they're both in the down position as we go across the whole molecule. Over here you have an up and a down position, so this is considered trans. And our enzymes fit this molecule, but they don't fit this molecule. So we can't break these down unless we're a ruminant organism that have bacteria living in one of our stomachs. So the cow can digest cellulose because they're ruminant organisms and they have bacteria living in one of their stomachs. The gorilla, however, cannot digest cellulose. That makes sense. They're the closest relative to us between these two animals, correct? So they must add another sugar source like fruit to their diet. So you see them eating bananas a lot, and then they can get their carbohydrates that way. So I wanted to talk about this ruminant organism a little bit more since I've mentioned it a couple times. So an example of a ruminant organism is a cow. So he has actually several sections to his stomach. It's 4 different compartments, with one of them being called the rumen. And that's where he gets its name. So the cow is going to eat his grass, follow the green line as it goes into the rumen. And this is where those symbiotic bacteria over here, these guys, are living in his stomach, breaking down the cell walls of the plant. So then after they've done their work, it continues on to another compartment here, the reticulum, before it is regurgitated. So the cow's kind of like throwing up his cud. And then he's going to chew it some more now that he's got the insides of the cells that he can work on, before he swallows it back down, and it's going to go into the omasum and then finally into the abomasum before it heads out into the intestines. So that is a ruminant organism with 4 different compartments to his stomach. So that's all I have for you today. We talked about carbohydrates, we talked about naming carbons according to their position in an overall molecule. We've talked about isomers. We've talked about different types of glycosidic linkages. We've given examples of monosaccharide, disaccharide, oligosaccharides, and polysaccharides. We talked about their functions and form, and how their form determines their function. So that's all I got for you today. Thanks for listening.